Okay, hi, so welcome back to the second video on microscopy. So what we're going to do is we're going to carry on where we left off in the previous video. If you haven't seen that one, then please do go and have a look, because we introduced calculations that you might come across in your exam regarding microscopy. Alright, and we're going to carry that on, and we're going to introduce something called standard form. Okay, standard form. Alright, now this topic does cross over from physics for one, and also from maths. You learn in your maths GCSE, I'm assuming most of you are probably taking maths GCSE, you learn about standard form. Right, and it's a way of writing very large or very small numbers in a simple, easy to understand way. You do it in physics as well, because in physics, obviously, sometimes the numbers are massive. If you're talking about like the mass of a planet or something like that, or the distance between planets, those numbers are going to be huge. Or if you're talking about the wavelength of something like light, then those numbers are actually tiny. And so if we're going to comment on those numbers or quote those numbers in meters, then those numbers are going to be either huge or really small and have loads of zeros in them. And so standard form is a better way of writing it. So let me give you an example. Let's say I had the number 3 million. Okay, so 3 million, that's a 3 and 6 zeros, and let's say that was meters. Right, now there are a few naming conventions, there are a few ways in which we could quote this number more simply, right? We could, for example, call that 3,000 kilometers, right? We could call that 3 megameters, right? But if we wanted to quote it in meters and keep it in meters, then we need to use something called standard form if we want to avoid writing all these zeros. And the way that we do that is basically you look in your number from left to right for your first non-zero number, right? Now here, the first non-zero number is clearly the 3, okay? And then that is where you need to put a decimal point after, okay? So the decimal point will come after here, right? And you'll get the number 3.000, etc., etc., right? Because importantly, for standard form, the way we write it is we have a number that is between 1 and 10. Okay, the number must be between 1 and 10, and the number 3 is obviously between 1 and 10. So what I would write down is 3 point, and I could write all those zeros if I wanted to, but I'm just going to write 1 0, right, to make it simpler. Now, that's 3.0 times by 10 to the power of something, right? And we put a power up here. And now there's an easy way to work out what that power is, okay? So if this is where I've put the decimal point up here, right there, well, the end of the number is where the decimal point should go, and that is here, okay? And so I'll do this in a different color. What I'm going to do is I'm going to count how many spaces I need to move that decimal point in order to reach where it should get. And that is 1, 2, 3, 4, 5, 6. And you see now it's where it should be. And so that requires 6 jumps. And so my answer is 10 to the power of 6. Right, if you were to think about it in literal terms, what you've got is the number 3 times by 10 to the power of 6. Now 10 to the power of 6 is a million. 3 times a million is 3 million, and that's the number that we started with. Right, so we've got 3 times 10 to the power of 6 meters. Okay? Alright, now the other one which does confuse people is when we have a really small number. Okay, but really the process is no different. I'll go through an example now. Let's say I had the number 0.000000018 meters. Right, so that's something very small, right? You're looking at the size of like a cellular component, for example. Okay, now, but this is the size, and we want to quote the size in meters. So how do we do that? Well, what we do, again, is we put the decimal point after the first non-zero number, okay? The first non-zero number, from left to right, is the 1, and so it goes after there. And so that's where the decimal point should be. So that number is now 1.8, right? You see there's nothing after the 8, so I can just leave it as 1.8. Okay, these zeros in front actually make no difference. They're not what we call significant. Okay, now what I need to do is I need to count the amount of spaces, okay, that I need to move from the decimal point to get where it actually is. And notice that we go backwards. Yeah, I'm going from right to left rather than up here, I went from left to right. okay, this way, whereas in this scenario, I'm going this way. If you're moving the decimal point to the left, then that is actually minus, right? We're basically saying we're going backwards, and so our power is going to have a minus. And so we still just count how many spaces we move. So that is 1, 2, 3, 4, 5, 6, 7, 8, 9, okay? And so that's where the decimal point ends up. So what we have is minus 9, and that is just the power that we raise 10 to. So it's 1.8 times 10 to the power of minus 9 meters. And that is correct, right? 1.8 times 10 to the minus 9 meters is the same as saying 0.000000018 meters. Okay? It's a lot easier to say, and it's a lot easier to look at. All right, so let's have a look at a kind of question that you may see in an exam. So here we go. It says a mitochondrion has a diameter of 0.0031 millimeters. State the diameter of the mitochondrion in meters, leaving your answer in standard form. Right, so there's two things here. You're going to have to convert the units, and you're going to have to use standard form. So please pause the video now and have a go at that, and then we'll go through it when you come back. All right then, so I hope you had a go at that. If not, then don't worry, because we're going through right now. So we have the number 0.0031, and that is in millimeters. All right, now I need to work out how many meters that is. Okay, and you need to know the conversion from millimeters to meters. A millimeter is 1,000 times smaller than a meter, or there are 1,000 millimeters in a meter. So that means this number must be divided by 1,000. Right, I always prefer to write it like this. Okay, either way, but this one is more mathematically correct. So if we divide that by 1,000, we get our answer in meters. Okay, and that is 0.000031 meters. Okay, so this is actually our answer, right? If it asks us to quote it in meters, then this would be our answer. However, it asks us to quote it in meters, leaving it in standard form. And so now we need to convert 0.000031 meters into standard form. Right, so what I'm going to do, I'll use a color again. Find the first non-zero number. First non-zero number is there, it's the 3. So the decimal point is going to go here, right? Let's just get rid of that. And now we count how many spaces do we need to move the decimal place for it to be where it started. And so that is 1, 2, 3, 4, 5, 6. Okay, 6 spaces. And it's 6 spaces backwards, right? And remember, that means minus. And so if we are now to quote our answer, it's this number here, 3, decimal point 1, times by 10 to the power of, and we move backwards, so it's a minus, and it was 6 spaces, so it's 6. Okay, 3.1 times 10 to the power of minus 6 meters. Okay, and that is our answer. Okay, right, so I hope that makes sense, because now we're going to have a look at using the light microscope, right? We spoke about it briefly in the last video, but now we're going to have a look at actually using it, because you guys are going to have to use these things in the lab at school, okay, to look at things like cells, normally plant cells. All right, so the first thing you need to do when you're using a light microscope is prepare a slide. And a slide is basically a flat piece of glass that is going to be put onto your microscope, and on top of it, that's where you're going to have the cells that you're going to be looking at. Right, so we need to prepare our slide. All right, so how do we do that? Well, the first thing you need to do is you need to add a drop or a droplet or a drop of water to the center of the slide, right? The reason for that is because the drop of water is where you're going to be putting your sample, okay? We don't want it to dry out, and it just allows you to be able to look at it through the lens, okay? Next thing you're going to do is you're going to actually prepare your cells, okay? So you're going to obtain your cells from somewhere. If you're doing this with something like a plant, normally you're going to be peeling a thin layer of tissue from the plant, right? So let's pretend we're going to be using a plant. We're going to peel a thin layer of tissue from the plant, okay? So that's all we're going to do. Next thing we're going to do is we are going to add the layer, and using tweezers, right? Because we're not going to use our fingers. Our fingers also have skin cells on them. And if we contaminate the plant cells with our skin cells, then it's not going to look very good. Okay, so we're going to use tweezers to add the layer to the droplet of water, right? I'll show you this in a diagram in a second. Okay, then what we do is we add a stain. Add a stain. Remember I told you in the last video that in order for us to see the cells, because cells pretty much are going to be just clear, okay, they're not colored, and so we can't really see anything. We add a stain and it makes some parts of the cell darker. Not the whole cell normally, but just some components of the cell darker. And depending on what stain we use, we're going to get different appearances of our cells. Okay, so we're going to add a stain. Sometimes a really common one is iodine. Okay, so iodine could be that stain, right? And that is going to be to our sample, right? So our droplet of water, basically. Okay, that will then soak through to the cells because our cells are already on there, right? Then what we do is we need to add what's called a cover slip. Okay, that's just a smaller piece of glass or plastic that goes on top of the water. Basically squashes everything down into a flat layer. It stops things being distorted and things from moving around. And most importantly, it stops things from actually getting onto your lens because you want your lens to be clean and clear, right? So we add the cover slip. On top of our droplet slash sample. All right, so that is how you prepare your slide. Okay, I'll show you a quick diagram of this. So, and this is obviously just made by myself. If you've got this at the bottom, right, this is your slide. Okay, it's just a flat piece of glass. This here is your cover slip. Okay, and what you're going to do, first of all, is you're going to add a droplet of water. Okay, so you add your droplet of water like so. Right, this is our water. We then take our plant tissue, right? So we've cut our plant, and this is obviously not accurate, right? This is not what plant tissue looks like. But let's say we've cut off like a little sliver of plant tissue. We then add that to our droplet of water, right? Inside that plant tissue is going to be cells, right? Tissues are made up of cells, okay? But we can't see them with our naked eye. We need the microscope in order to see them. Then what we're going to do is we're going to swoop in and we're going to add some iodine, right? Iodine is like a purpley black, so I'll just draw it purple so it's nicer to look at. And you blop some iodine on there, right? That will then allow us to see the cells because you've now stained the cells. Lastly, whoa, that's not what we want. Lastly, what we do is we add our cover slip to the top, right? You put your cover slip on the top. Now it's all flat. Right, that looks like a mess because I'm not very good at drawing, but you can see step by step what we've done. Now this whole thing, right, is going to be ready to move and place under a microscope. Okay, and that's how it works. Now you need to know what you're going to do with the microscope because it's not as easy as putting it under the microscope and boom, you have your great image. Okay, because it's not as simple as that, because it depends on how much we've had to zoom in, etc., etc. So what we need to do now is we need to look at a microscope, right, and discuss what we're going to do with it. All right, so let's run through quickly the different parts of the microscope. So first here we have the eyepiece. This is obviously where you are going to be looking, right? You look down it with one eye, that is the eyepiece. Here we have the adjustment knob. Okay, it's the adjustment knob. You do see these things named differently. Okay, but the adjustment knob, basically, you can twist it and it causes the the stage, which is this, to go up or to go down, right? So you lower it or you raise it up, depending on what you need to do. Okay, this here is called the focus knob. Sometimes you see it called the fine focus knob, etc., etc. That focuses your image until you get it so you can actually see the intricate detail, right? Sometimes your image will be really blurry. Sometimes you won't really see anything at all until you twist this, and then eventually you'll find that you can see everything. Okay, that's what that's for. Now, these are the lenses. Okay, these here are the lenses. You'll notice you have three. Most light microscopes do have three. Some may have two, some may have four. Right? But they are the lenses and they are basically what is going to magnify Okay, that's how you zoom in and zoom out, right? This here is called the stage. Okay, this is the stage. This is where you're going to clip your slide onto. You'll notice that there are two clips on top of it there, and that's where your slide goes. Lastly, it's called a light microscope because it produces light, and the light shines from the bottom upwards through this hole here in the stage and into the lens, and obviously eventually into your eye, and that is how you see. Okie doke, so let's jump straight in and talk about what you actually have to do. Because you've prepared your slide, we just talked about how to prepare the slide. Now we want to have a look at it, right? We know the parts of the microscope. What do we do with them? Okay, first thing you do is you clip the slide onto the stage, right? The slide must go onto the stage, and the middle of the slide, so where your cover slip is and where your sample is, wants to be at that hole. Let me just use a different color so I can highlight it. Wants to be here, okay, in the center, because that's where the light comes through. The light needs to go through your sample. All right, what do we do next? We select the lowest powered objective lens, right? These are called lenses, but they're sometimes called objective lenses, right? That's the correct name for them. And the reason we have more than one is because they're different power. They produce different magnifications, right? We want the lowest magnification first. So the one that's, they have written on them as well, times whatever they're going to be, maybe times 50, times 100, okay? And we want the lowest number to start with, right? Okay, then what we do is we use the adjustment knob, okay, and to move the stage, okay, so where the slide is sitting, just below where the lens is, okay. Use the adjustment knob to move the stage upward to just below the lens. Okay, that's so you can get a good look at your sample. Okay, then we're going to actually have a look. Okay, now we look down the eyepiece. Okay, now at this point, you may have zoomed in too far. When you've got the slide just under the lens, you may have zoomed in too far. Okay, but so now while we're looking down the eyepiece, okay, we then move the stage down with the adjustment knob until the sample is roughly in view, right, or in focus. Okay, it might be really blurry, but you want to be able to just see the outlines. Okay, then what you're going to do is adjust the focus. So now we use the focus knob to adjust the focus until there is a clear image on the slide, right? So when you can actually see something clearly, that's when you have focused it correctly, right? Otherwise it's going to be blurry or you're not going to see anything at all. Okay, and that's basically it, unless you want to zoom in even further. Okay, so if needed, then swap the lens for a higher powered one and refocus the lens, right? So when you swap the lens, now your image may not be focused again. Now you're gonna have to refocus it, and this may be using the adjustment knob, but it's more likely using the focus knob. You may have to do both. Okay, it's quite it takes a little bit of getting used to, but once you're used to it, then it becomes second nature. But that is all you need to do. All right, and so lastly, you are often asked, so when you're doing these kind of experiments where you're looking down a microscope, you're often asked to make drawings, right? You want to draw what you can see, and there are important things that you need to do, right? I'm not going to dwell on this for too long because it's quite straightforward, but basically what you need to do is you need to draw with a pencil. Okay, here we go. So your drawings must be with a pencil. Okay, so drawings with a sharp pencil, that's important as well. Okay, what we need to do is take up at least half of our drawing space. Okay, that's just so that we have a large drawing, right? If they gave you an A4 sheet of paper to draw and you drew a tiny drawing in the top left-hand corner, then there's no point. You've basically wasted all the rest of the space, okay? And you're going to miss out some detail. So you take up at least half of the drawing space, okay? when you're drawing, you need to use unbroken lines. Okay, do not color or shade. Okay, we just do this in pencil. We draw the outlines, but that is it. Draw outlines of features seen and label them. Okay, for example, I'll put here, e.g. the nucleus, right? We may have to draw the nucleus and we'd label it. We still wouldn't color it in though, okay? You'd draw it, you would label it, but you wouldn't color it in, okay? So we also need to write a title, and this should be a separate point. Include the magnification, okay? Magnification on the lens used. Okay, this is so that we can calculate the size of the real image, right, or the real object. If we know the magnification, okay, then we can calculate the size, right? So as a really quick example, I'm not going to do any calculations here because you actually need one more piece of information. But let's say I was drawing a cell, maybe, and I only had the space to the left of the writing here to do it. Let's say I was drawing a cell, I might draw something like this. Notice it's taken up more than half. There may be a nucleus here, right? That's in there. The lines are unbroken. I haven't shaded anything. There might be a mitochondria, right? Might be another one. Might be another one. Okay, and that may be it, right? So then I may label it and say that this here is the nucleus. Okay, and then I might say all three of these things are the mitochondria. Remember, you don't have to draw the cell completely. If you can see the mitochondria, great. You don't have to draw every single one of them. You just need to give it a rough idea. Okay, and that may be my diagram, and that would be perfectly fine as long as I didn't shade and as long as the drawing was big enough on the sheet of paper. Okay, so that was quite a lot of information. I'm going to stop there. So please do comment if you do have any questions, or feel free to send me an email using the link below. But do like and subscribe because that does really help me out, and also it will notify you whenever new videos become available. And I look forward to seeing you in the next video.